Okay, so today we are going to finish our chapter on gene expression. Now, we're going to go over some mutations today and different things that could happen, but before we get to that, so bacteria and eukaryotes, they will have multiple ribosomes translate mRNA at the same time. If we just did one, it would take a while to make our polypeptide chain. But if we have a bunch functioning at the same time and we have all these ribosomes, why not use them, right? This will allow our protein to be made a lot faster, and then it can go do whatever its job it needs to do. So we can connect these ribosomes together, and they're called polyribosomes. And here's a picture of one here, and that's under an electron microscope down there. But you guys can see that there is more than one ribosome, and given this, we're able to translate our mRNA a lot faster. Okay, we're good there. Now here's just a kind of summary of everything we went over, guys, with transcription and translation in one picture. So we have the transcription inside of the nucleus. We have the mRNA leaving the nucleus going to our ribosome, and then it transfers from the A to the P to the E site, where the tRNA exits at the E site. The P site, we get the polypeptide elongating, and the A is just to make sure that everything is good with our connection between the mRNA and the tRNA. So now let's do the mutations. So last chapter, I believe we went over chromosomal mutations. These ones we're going to go over nucleotide or DNA mutations. So in the last chapter, when we talked about chromosomal mutations, this were a whole part of a chromosome was deleted. So a lot of traits were deleted or duplicated. This one, we are going to go to finer mutations where we're just dealing with a very small portion of our DNA. So the first one we're going to go over is called a point mutation. This is where we will have a single nucleotide in DNA mutated. Maybe we had an A there and it gets mutated into a T. So what can happen here is, if I move myself somewhere, I don't know where, but there we go. If you could see here, we have normal hemoglobin over here, and over here we have some kind of mutant hemoglobin. If we have our mRNA, which goes off of the CTC, would be our template strand, the GAG would be our coding strand up here. But the CTC, we want it to go to GAG. Over here, instead of it going to GAG, it goes to GUG, which completely changes that amino acid that it gets translated for. That's a point mutation. There's different types of point mutations that we're going to do. There's also, we can do a insertion or deletion, and we'll talk about these in a little bit, but they are a little more severe than just changing one amino acid. Oops. Okay. So the first one we're going to go over, the first point mutation is called a silent mutation. And I'm going to use my chart here to show you what a silent mutation is. And I'm just going to use my mRNA to start off, instead of going from DNA to mRNA. So silent mutation, let's say our original mRNA is, let's do C, C, U. And then that mRNA gets mutated to C, C, whoops, CCG. All right, CCU, let's use our chart. CCU, CCU goes to proline, or PRO. CCG also goes to proline. This is a silent mutation. We did have a mutation in our base pair, but it still translates to the same amino acid, so no harm. Okay, so silent mutations are probably the best type of mutations you can have because it keeps that amino acid sequence the same. And here's another example of one. All right, here, okay, same example I went over, just a bigger sequence. Second one is called a missense mutation. So I'll come back to this to go over nonsense, but let me show you what a missense one is. So missense, I'm going to do it the same way I did last one. So let's do mRNA. Let's do GCU. And then let's get the lights back on. There we go. Let's say the second one goes to, let's do GUU. So our C is now a U. That's our only difference. GCU goes to alanine, ALA. GUU goes to valine, VAL. So with that one change in our nucleotide, we now have a completely different amino acid. So that is our missense mutation, where the amino acid does change. Second one is called a nonsense mutation on this slide. This is where we get a premature stop codon. Now, that stop codon will trigger that polypeptide to stop forming. So we are going to have a shortened polypeptide chain, which that protein is not going to be usable. So let me show you that one, what it looks like here. So we're going to have our mRNA. And this will usually be in the middle of a sequence somewhere, guys. So let's say that we had UGC, and then it goes to UGA. So UGC would go to CYS, and then UGA would go to our stop codon. And like I said, guys, this is going to be, if this is our mRNA, this is going to be somewhere in the middle of our sequence. So all of this stuff afterwards, after our stop codon, is not going to get translated. We're going to have a very short protein sequence. All right, so we're good there. So those are our three types of point mutations. We are just changing one nucleotide for a point mutation. It may trigger a change in our amino acid where we could get like a missense one, we get a new amino acid. We could get a nonsense one where we get a premature stop. And we could also get a silent one where nothing changes. Now these are the bad ones, the frame shift ones. What happens in a frame shift mutation is we insert or we delete a nucleotide. So let me show you this here. So our top one here, our top blue one, is our coding sequence. So we have T, A, C, T, T. That's all I'm going to go. Look down here. This is where our mutation occurred. We have T, A, C. We inserted this A, so that's an insertion, and now we have TT right after it. What this does, guys, is up here we have TAC going to AUG for our mRNA, and it goes to methionine. TTC goes to AAG for mRNA, it goes to lysine. Down here, that one insertion changes every single amino acid past this. This would go to TAC, which, okay, it's cool there, everything's good, but inserting that A completely changed our reading frame, where we have UAA for our mRNA, and that does do a stop codon, but if we even look further, this one should be CAA, now it's GUU. This should be ACC, now it's UGG. It changes everything up, all right? So it's not good. It's not what we want. And if you compare that up top, it does completely change our reading frame. This one is a deletion. So up here we have TAC going to methionine. Everything's good there. Everything's good there. Right here we have AAA, and we deleted that last A. So instead of it doing AAA, now it's AAC. instead of doing CCG up top, it's now doing CGA. Okay, so everything gets shifted over to the left one. All right, and like I said, these are the worst guys just because it completely changes all of our amino acids. Okay, so how do mutations occur? A lot of different ways. Sometimes just, hey, stuff happens, right? Sometimes our DNA doesn't replicate properly. Maybe at one of our checkpoints, the proteins that regulate those checkpoints, whether it's during G2, after the DNA has been replicated, maybe that one's not functioning properly. Maybe during the metaphase checkpoint, the proteins weren't working properly, the ones that were functioning in the middle there, to make sure all of our spindle fibers were attached. So that way, we get chromosomes going every which way. But regardless of this, guys, something happened, okay? It could even been DNA polymerase wasn't working properly, all right? But these are things that can happen in our body every day. We hope that if it's, DNA polymerase 3 just misses it, we hope that something else is going to catch that mistake. And if it doesn't, most of the time it's not going to be with cancer, or it's not going to result in cancer. Every now and again it does, but, most of the time it doesn't. Now, there are some different things we can put in our bodies to kind of help the mutations become more prevalent, or our DNA to mutate more often. Things like, all the carcinogens in cigarettes and stuff like that. People put that in their bodies, and then they wonder why, their DNA mutated or why they're getting cancer. All right. So some things, cancer is from our bodies. You might not have smoked a day in your life and you get lung cancer. It's like, what the heck happened? All right. Some people, you might smoke till you're 100 and not get cancer. It's just sometimes our bodies function properly, sometimes they don't. But it has been proven that carcinogens do increase your risk of cancer. Okay, guys, that is the end of the chapter on gene expression. You guys can finish your reading guides for that. And have a good rest of your day.